Thank you everyone for being here today. It's a great pleasure to have Joseph Hibdon giving our seminar series talk. Joe is a professor at NEIU, just north of us here, and is currently the rotating associate director for convening activities here at NITMB. So it's really great to have you speak, and I'm really excited to learn more about the work that you've been doing.
All right, thank you so much. I was very strategic about the placement of when I'm giving this talk. I was hoping you all might be traveling for Thanksgiving soon. It's interesting. One of the things that I always do is when I'm thinking about my talks and what I'm going to talk about, the most nerve-wracking part is thinking about the title. And I actually don't like this title. And all the time when Tuka comes in and welcomes people to NITMB, one of the biggest things he talks about is biology inspiring mathematics. So I should have come up with a better title. And I should have said it's biology inspiring mathematics for undergraduate research. I think that is a way better title. So if I give this talk in the future. And this is going to be quite a bit different talk than what we usually see here at NITMB, because I'm at a primarily undergraduate institution where my research is strongly related to the ability of what my students can do and the ability for me to engage my students in that research. So there's a collaboration involved. So what am I going to talk about? The talk is broken up into three parts, but we can think about it as two parts. One is more an ecology focus, where I'm talking about cattails and some very simple model related to invasive species. And then I'll go into my more recent work with students is based on working with Myxobacteria, in particular doing some very simple agent-based modeling. And then finally, the work that we did over the summer, which is working with large language models for cancer drug discovery. And one of the things that I've picked up actually coming to some of the talks is I should start with acknowledgements, because we all forget that, or if we run out of time. Here I'm just acknowledging some of the people I've worked with or engaged with. For the cattail side, I haven't worked directly with Pam Geddes. In terms of this, we work on many other projects. But I've engaged with her students who have been doing the research for 10 plus years, where they go out into the field all the time. And this is something, as a mathematician, I don't do, right? I garden. That's my field work. But these students have been doing this for a long time, where they're investigating these cattails. However, I've had the fortune of working with two math students, and my MS students didn't want their names. My former master students didn't want their names on here, so I didn't put their names. But we've taken some of their observations, and we'll talk about what that means. In terms of the Myxo work of the Myxobacteria, some of you have met Amina Stojkovic. She's been here before. She's worked with many biology students, and including high school students and chemistry students. And Ken Nicholson, who's in the chemistry department at NEIU, who this last summer worked with 3 graduate students in chemistry. And then I have my list of the math students, and we'll talk more about them as we go on. And I am somewhat of a hypocrite from the way I teach my students in the way that they should be presenting. I'm going to be telling a little bit more of a story. And I did my PhD in fluid mechanics, focusing on combustion. And obviously, when you're going to a primarily undergraduate institution, you can do a little bit of that work, but there is a much bigger hurdle of engaging students. And one of the things is I had the opportunity to work in the Student Center for Science Engagement when I was first there. And most of the funded research was through the U.S. Department of Agriculture and the Department of Ed. And many of the people doing the research was focused on the biology side. And so you're sitting there always talking to biologists and to chemists about what they're doing. And then you get to see the students engage with the students. And that engagement really shifted the way I've done my research. And one of my favorite things was looking at these cattails. And what was happening is we have latifolia on the left here. This is a native cattails that are throughout here in the Midwest, and we see them all the time. If you're driving up north over at the Skokie Lagoons, you'll see them. A little bit ago, you should have seen them start to flower. They have this beautiful flowering where they pop, and this spreads the seeds, and they push into the wind, and they continue. Some interesting things is they have very important historical uses for the Native American population in the area for medicine, clothing, food, and art. So historically, it's embedded in the Midwest here. And then more importantly, they provide an ecosystem for native species, wetlands. And we know that wetlands, we can see the shifting of the wetlands that helps with erosion. There's a lot of important pieces to maintaining these wetlands. On the right here, we have this, and I know I was going to mispronounce it because I get teased by my students all the time, Dominogenesis. It's a native to temperate and tropical regions, much more further south. So they need a warmer climate to be maintained. Now you'll see something interesting here. They look very similar, don't they? If you were out there, you'd be like, oh, they're the same. There is some morphological traits that you can pick up. The male part here up top is a little bit bigger. They're a little bit denser, but typically they're very similar. However, because they're denser, they do affect the way the wetlands and the native species of insects will be able to live in these wetland microenvironments. So what happens is, unlike normal invasive species, when I think about it, I always think about the classic squirrel problem in England. And I mean, that's how we teach it. Every time it's like, oh, there's these squirrels, they got embedded, and then the more aggressive squirrels beats out this is a very famous problem. But we don't talk about what's called crossbreeding, where we get this hybridization of the species. So what has happened is, as this invasive species comes in, it's not like they completely take over the environment. What they will do is they will start to crossbreed and they will form a new species. What's more surprising, and this is relative in the last 5 or 6 years, is it doesn't just form one, it forms many different species. And each of these different species have different characteristics and different traits. And actually you can quite see here that these are very distinctively different. But it's not always true that you can identify that they're distinctively different. So I guess, what is the issue? So one of the issues that we have is that this hybrid species is even more aggressive in terms of they will come in and they will grow and they will take the rich environment and they will overtake it. So aggressive, you wouldn't think of like a squirrel being aggressive. Aggressive is more as they take up the nutrients. And they'll push out not only the native, but they will also push out the invasive species. It reduces other native plant biodiversity quite rapidly. And you can imagine if you have small grasses that are there below the cattails that are trying to impede your erosion, that's a big problem. And we already see it's a problem. Again, if you go up to the Skokie Lagoons, you can see this erosion. They're very hard to eradicate. And the reason is it's morphologically hard to distinguish between the other species. Not only is the native species hard to distinguish between the invasives, I can't go out there. I'm not an expert, but if I tried, I'm like, let me help out. Let me see if I can help you, because we do this in prairies all the time. I can't. So not only the experts can't even go out there and say this is the invasive and this is the hybrid. Sometimes you can. And more importantly is there are some molecular tools that are being developed for them to identify what is the native versus the invasive and then these hybrid species. But they're very expensive. So the lab, Pam Getty's lab at NEIU, was to create these cheap, relatively inexpensive ones that can be used quickly. But are you going to do that for 1,000 species or 1,000, plants in a 20-foot? No, it's not going to happen. So there's a lot of problems related to this. So they've been working for a long time identifying these species. They have developed these lab tools for these biomarker identifications. But the question is, I see this, and as I mentioned, I'm like, oh, this is a very quick, fun, invasive species problems. I can give it to a student, go play with it. And of course, that's what we always do, isn't it? Okay, it takes every ounce of energy for me not to sit and do it myself. But that's part of being at a primarily undergraduate institution, is to pull back and to sit back and say, you play. A few things. There are many invasive species models. They've existed, and you're going to see the problem that the students came up with. Relatively simple, and you're like, oh, of course this works, but there's nothing in the literature that says where we have this creation of a hybrid species. And that's kind of the unique piece. And these are relatively easy enough for a master's student to play with and to have many questions to explore and to continue to go with. I've had a few students play with, but actually the most promising was Wasal Hamo, who's actually a former NITMB SERP student. And what she did was she was here for the summer. She took the skills that she obtained from I think it was working with Bill here, and then brought it back to NEIU. I was like, oh, I can actually really dump this on you and play for the year. And we had some Department of Ed funds, and I'd be able to fund her while she was taking courses. And most of my students, she was working full-time or working part-time at a restaurant, and was taking a full load of courses, and she could drop her work time to be able to work on this problem. And her first attempt was this very simple model. It's straightforward, good dynamical systems. Notice I'm avoiding PDEs. I'm not worrying about the distribution over space, because typically students don't have that at the undergraduate level, and that's fine. The native and invasive, this is from a paper in 2017, and they have a few more terms, but we tried to simplify it down. Again, playing with the gray squirrels. I think people have been playing with that for a very long time. You can play with that for the rest of your career. And then she added this hybrid species. And one of the things that's quite unique here is, we have this, classic logistic growth rate, and this is this competing forces between the native and the invasive species. But the hybrid species is like, hey, I'm just going to grow. Once I've been created, right, I'm just going to grow up to whatever the carrying capacity is. You'll notice that we're assuming the carrying capacity is the same across all three species. That's a very bad assumption, but it's okay. It's a start, right? Keep the model simple. And she's like, oh, okay. She's like, what should I do next? I'm like, well, what did you do over the summer? She's like, well, I graphed it. It's like, okay. And it behaves. So if I solve those equations, this is the behavior I expect. I expect because of that plus gamma term where we're having the two species interact, the native and the invasive species, I expect this to grow very quickly, and then because of the competing forces to drive down from the carrying capacity of the logistic growth rate. So it has the behavior. And obviously, the next question should be, well, did you do any stability analysis? And she's like, I remember that term. I remember some of it. What do I do? Again, this is a student that, yeah, please interrupt at any time.
If you ran this for another 30T in time, do you eventually see the hybrid species kill off the invasive species?
Yeah, so that was my question to her. That's a good question. And that's what I said. How would you know, right? You're always looking at the extremes. So I'll answer that in a second. Because then she went to this. And again, all of this is actually pretty consistent with what you see in the literature. I'm curious about this piece. And I put the word in progress because every free moment she has, the equations seem simple, but when you're sitting down without any numbers and you're just playing, Um, for an undergrad, if you don't have dedicated time to do it, and you're taking a full load of courses, still taking care of family, and I could sit down and solve this for her. That's not the issue. The issue is getting her to do it, right? And for those that my colleagues here that have grad students and postdocs, I give you a lot of kudos because my patience is very yes.
Basically have everything dies, everything goes to one state for the next three.
Yeah.
There's a balance between two states.
Yeah.
And there's a balance between states. I just want to make sure I'm reading.
Yes. Hold on. Yes. And she actually has this part right here. So we do know that there is a steady state solution, but we don't know about stability. Based on the equations, I'm assuming it's not stable, but I don't know. I want her to prove it. I want her to show it. I don't know for sure. So yeah, so let's talk about going out to 30 to further. So she adjusted the parameters. Now you should be asking yourself, what about data? Where's the data to help put into? The problem is the data is not very good. It is terrible, actually. So yeah. So that kind of answers your question. They do coexist, but you have to be very careful with what the values are for coexistence, and we see that in this steady state chart. Yes. Yeah. Go ahead, Cody. Yeah, please interrupt at any point. I actually love questions. So that was happening up until some funding cuts to the USDA. So yeah. That's exactly that is the proper lab approach. Yeah. Well, go ahead, Alistair.
Can you go back to your tape?
Yeah, please.
So if this condition for stability is one of them always satisfied? No.
Yeah, so this condition is only satisfied if this criteria is maintained. And so what you're saying here is that the growth rate has to be quite large, which makes sense. Yeah, if it exists. Yeah. And I'm guessing actually based on some preliminary data that's seen that they do have that this does not exist.
And ours are growth rate of base for the
So RU is which makes sense. Yeah, no, yeah. Sorry, I'm being very quick to tell the story, but yes, that is correct. Which and that was our conversation too. I'm like, oh, good, it makes sense. Otherwise, we'd have a little bit of an issue. But again, the equations are relatively simple, but you get some interesting, readable dynamics for an undergrad. Go ahead, yeah, please.
If all the growth rates are small enough, then I maybe would expect to get.
Yeah, that's what I'm assuming. Mm-hmm. That would be my assumption as well. But she needs to prove it first. Yeah. Yeah, I mean, so the model itself doesn't take into account that it's a cattail. It just takes into account that you have some crossbreeding and that it is necessary for the species to even start to have this crossbreeding. I don't know any animals that do this, but I'm guaranteed there is. I'm not, I don't have the exposure. There probably is. But every animal model doesn't talk about hybridization. like everything else in life, every model, there's many issues. I mentioned about the data. What is really happening in nature about these growth rates? These growth rates do change. Notice that the carrying capacities were the same. They're definitely not. You'll see more with the hybrid species. The carrying capacity is much higher. You can have a higher load on the environment. We want to expand into account if you have multiple species. My first thought actually was a delayed differential equation, but delay the start for a while. And so there's many other options that we can take into account here and take into effect climate change. How about the student results? I had a couple students graduate playing with this project. Basal presented a poster at AWM National Conference in 2025 and that was her first national conference she ever got to present at, and she had a lot of fun and good feedback. On a personal side, on the left here, I spent 2.5 years working with the village of Skokie on redesigning the seal, and you'll notice that there's cattails in there. So I was inspired. But it was interesting. There was a lot of pushback from some of the people that lived there that said, aren't cattails an invasive species? I was like, oh, I have a whole story for you. And I worked together for us to, have this story of understanding that there are some that are invasive and some that are not. And we talked about the historical importance to the native population as well. One other thing, I spent, for 11 years, I had a MARC training grant, an NIH MARC training grant, which funded students who are interested in going on to do PhD programs in biomedical sciences. I worked with Amina and a psychologist, Brecky Church, that explains, for those that were here earlier, I was talking about behavior and learning dynamics, and that's what her expertise was in. And I got to mentor students from biology, chemistry, psychology, and environmental science. You'll notice there's no math students on here. There is at the very last year, and then the funding was cut. And one of the things that I learned from this experience was how to communicate and talk with biologists and psychologists, and how different their world is compared to mine as a mathematician. I struggled already in the beginning with the students, but where you're really sitting down, and we would have meetings with these students once a week talking about the research that they're doing from a very broad spectrum of disciplines. And you realize, well, I realized pretty quickly is, one, I feel like I'm in over my head. But two is my skill set is to talk to them about the quantitative side. I can help them with their quantitative analysis and how you report it properly. But the way you report it is very different based on the discipline. So I struggled for a long time. But one of the students was presenting something, and she brought this picture. And what she was doing in the lab was she was growing these bacteria, these soil bacteria. And she gives this, and I was like, wait a second. I turned to her mentor, Mina. I was like, is this real? Is this a fungus? It's not a bacteria? Because I see this, I think of a fungus right away. And she was like, yeah, I had her do this multiple times because I didn't believe what we were growing. Yeah, this is actually just an image of an agar plate. So it's quite large, what you're seeing. And we have the following. So she put up that constant dark one, and then she put up this one. I was like, and the student looks at me, she's like, I don't know how to explain this. And Mina then looks at me. I also don't know how to explain this. I was like, I think I can model the left one. Maybe. I mean, my first instinct is to look at is it spreading, right? And you have. So what is this? What are we seeing? So what this is on the left here is these dots are. So what they're doing is they're plating these bacteria. And over time is the bacteria are collecting together and they're forming these multicellular structures, which are called fruiting bodies. And that's what these dark stains are, is the fruiting bodies. And you can see them. I mean, if I had I wish I asked to bring a plate for you all to see, but let's just say that didn't go over very well. Yes? So the very beginning, the right here is a human, and we're going to see a future diagram. But the growth pattern that you're seeing here is what it's doing naturally. And we're going to try to understand why. Yes? It does, but these are soil bacteria, so they're typically under the ground. And so that's what this 12-hour light dark cycle is showing. We're still seeing some growth of these fruiting bodies, but the question more is how can we impede the growth or how can we enhance the growth? I think is a better question. Yes. Yeah, and I mean, my first instinct is just pure diffusion. I mean, I see this, I'm like, okay, it's diffusing, like from the center and it's moving out. But I guess the question is, why is it not doing it as uniformed over here? It could be the light and the growth impeding it. Come to find out, there's a lot of dynamics here that are not known. Yeah, go ahead, please. Oh, I do. I have one. That's right here. You're cheating. No, I'm teasing. No, but that is the question. Yeah, I don't have a movie, and there's reasons why. One is at an institution like mine is we don't have the capacity to actually do movie escapes for very long times. I think we do now, but at the time that this was first done about 7 years ago, we didn't. So it was actually my first question is I want a movie. That is like the first question I had too. It's like, can we get a movie? Can we please just put something over there and watch it? Or can we get some highlights of these bacteria as they're moving and have a movie? Because that can answer a lot of questions. Yes. Yes. Yeah, for this one it is. Yes. Yeah, and that's as soon as you do that, you're disrupting. So this is not something we'd say this is completely dark, but this is mostly dark, except for when you get the one image. So you can see in the 4 days, we have this formation of these fruiting bodies. That's what the darkness is. They put down a bunch of bacteria right here in the center, and this darkness is the collection of these fruiting bodies, or the white. After 7 days, it looks like that the bacteria, my first guess was that the bacteria is just spreading. Some of them are forming, collecting together to form these multicellular structures. And we're just getting it based on diffusion, pure diffusion. And you can see over 14 days, it goes pretty quickly. Oh yeah, because this is the same one. This is the same plate. Yeah. Yes. Yeah. That's exactly what it's doing. Yeah. And I have an answer for you towards the end of why, but we didn't know it's first why. But yeah. Yes. Yeah, it could be. And I at the moment I think that's what we're thinking. But it was my thought. I mean, this is somebody that worked with flames and diffusion flames, so I was like, oh, diffusion. It's always diffusion. It's cheating. It's a cheating answer, but it's actually not. Yeah. Yes. Yeah. Yes. Um, we don't know. We don't know if it's even spacing because this is one experiment. And when they run different experiments, we don't see it as clear. This is probably our best experiment that we have of the data. Um, I thought it was even spacing, but we find out it's not necessarily even spacing. And it really is environmentally specific. And it's quite sensitive. Yeah. Yeah, and that's what it is. That's actually exactly what's going to happen. Yes.
Tell me a bit more about like
Yeah, I'll show you. Yeah, I'll show you the whole fruiting. I'm kind of like giving you the flavor before I get to tell you the full biology mechanism. Because the biology I find even more interesting than these beautiful images. One of the best things that you can do as a mathematician is I just sit down. I'm like, okay, I show this. I have somebody else verify it, and we play. So what happened was Amina took these results, presented at a Gordon research conference, and was saying like we had all these questions behind it. And a colleague of hers from UC Davis like, there's no way. Who studies Myxobacteria? That's his research is. So he had a grad student reproduce it. And what we have here is, here we have pure agar, where you have almost no nutrients in the system. Okay. And all we do is we don't see any of that spreading. Okay. So it appears that nutrients are required. Next, we have this standard agar, which is, we would call this a limited amount of nutrients in the system. And we see this spread. We see these concentric ring formations of these fruiting bodies. And then if you have a lot of nutrients, you see fruiting bodies, but we don't see these beautiful concentric rings. So nutrients is very important. So that will come to the biology. But I guess my first thought was like, hey, why isn't it just like a fungus? Because this is what a fungus does. A fungus does this. If anybody studies this, they've seen this image probably on the left before, where you're looking at the circadian clock of fungal growth. And some of the biologists that Amina was talking to, they're like, that's probably what this is. It's just a circadian clock similar to a fungus. And you can see if we do this with the Myxobacteria here, similar type of behavior. So there could be a circadian rhythm that's happening. Come to find out, actually, there is a circadian protein that's in these Myxobacteria, and it goes back to that they're soil bacteria. So what have they been doing? So Dr. Stoyevic's lab has been working on this for many years. And again, I talk about that there's soil-dwelling bacteria. They're everywhere. And we're going to come back to the importance that these are everywhere. So let's talk about this life cycle. So this answers your question more about what the life cycle is. Typically, actually, when I do this, I actually start with the life cycle. But I think the story of like students presenting these with limited knowledge, like all these questions that you're asking, that we're asking here, is the exact questions I was asking. I was like, I think I had like 2 pages of questions for them of like, why is this happening? Can I get a video of what's going on? Can we actually make the bacteria darker so I can actually see what's happening with these bacteria as they're moving? So what's happening with these Myxobacteria is we have these vegetative cells. And these are what's typical when you think about bacteria, right? They're just moving around. Eventually, over some time, they will aggregate and cluster together. And this aggregation and clustering is caused by some environmental factors. As we can see in the last slide, looks like nutrient dependent. They will then collect to form a mound of cells, and then they will form these fruiting bodies. And these fruiting bodies, what's happening is the bacteria is actually switching behavior. And they cluster together, and now you have a multicellular structure. So you went from very simple, relatively simple. I have to be careful. The word simple for bacteria. Those that study it don't like that word. And to something quite complex with a fruiting body. These fruiting bodies then will shoot out spores, just like a fungus does. So that explains a little bit of what we are seeing, why there's a relationship between what we see with a fungus. These myxospores then will germinate, and then they'll grow, and now we have our bacteria again. And you can imagine, so if this is a nice little scan of what these fruiting bodies look like. They form the fruiting bodies, and then it's spitting out the spores. And those spores then spread. And that may explain why there's that distance. Yes? Yeah, and that was my thought. And I thought it was like a chemically induced, right? So if there's a chemical induction that's saying something behind them where the bacteria is saying there's a signal, like my bacteria is here, and now they've coalesced. And it's like, well, we know there's nothing here. We've coalesced in this fruiting body. Well, there's no signal here. I'm going to shoot the spore this way and try to hunt for more food again. And that's actually what I thought was happening. I still do. So there's a. So okay. So my math part comes in, and I said, okay, let's model this. But we're going to do it a little bit differently. We're going to do an agent-based model. So I'm going to treat each of these bacteria individualistically, and see if we can just even get the concentric rings. I don't want quantitative agreement. I just want qualitative. What are some of the simplest assumptions that we need to make to be able to get this, like behaviorally for the bacteria? Yeah, I don't know. I think you can, because I think it's just distance. It's like a distance of jumping. Yeah, and I did give that to a grad student to play with, and it didn't go well, but it could have been the student. It could have been me directing. It could have been me guiding the student. I don't blame it on the student. I blame it on myself as the mentor. But yeah, I should be careful. Actually, yeah, because I actually like that approach a lot, because okay, I'll just go this way, and then do we see the spatial jumps? And we use that logo. Simple. It's through Northwestern. I don't have to worry about coding in Python, or the student does. And I teach a course actually which uses it. So the first student did this. It was Jerome Williams, and I want you to notice the year. The summer of 2020. We were in complete lockdown. So the plan was I wrote a small mini NEIU grant, and I was like, okay, I want them to be in the lab. I wanted them to go into the lab to play, to see what was happening, and help that guide the assumptions in the development of this agent-based model. Obviously, that did not happen at all. I think we were one of the few people actually doing research over the summer. But he came up with something very interesting. And what he decided to do was he said, okay, I'm just going to have them plate the bacteria, and I'm just going to have the bacteria move randomly, but wanting to move away from the center. That's it. And have just after a certain amount of time, that assume that they're going to coalesce to form fruiting bodies and move on. And that does work. But of course, we're forcing it, aren't we? There's no real biology except for move away from the center. And so I was like, okay, that's way too simple, but we get some quite interesting results. One of the things that came up was in the lab that we were like, what happens if you start to plate multiple ones? Are they impeding the formation of the rings between them? And I can say that maybe there's something going on with the chemical sense, and saying, okay, don't come to this area, please move away. If that distance that was being asked shrinks, that could be also indicator of some chemical response. This has my millimeters. This is 2 millimeters. So this is zoomed in onto that plate. So it gives you some sense of scale. Question? Yeah. Please ask questions. Like I appreciate questions. Not in this one. Not in this one. That in the next one it is. And because we come to find out, if you saw those previous pictures, that was from our collaborator at UC Davis, Mitch Singer. It is nutrient dependent. I mean, that we kind of knew that was true. But at the time in 2020, we didn't have those results. That picture that I gave you was actually in 2022. So all I did was just movement. That's all I did. Yes. Yes, I'm almost positive that's why. So I had a more recent student who's a physics student who was Mark student, McNair, and honors. So this is like one of the best students I've ever had at NEIU. They've all been great, but like he was very good about just like diving into the problem. You know, we always get the student that just wants to just put everything they have into it, even though he's a physics student and interested in quantum mechanics. So what he did was he's like, okay, what if it is chemical signal response? So he took the agent-based model. He's like, allow for that spreading of the movement, but he's saying we're going to have a chemical signal response. So what's happening is we're assuming no matter what always happens, we have sporulation, or we have fruiting bodies in the center. They're going to sporulate and send out. Those spores release a signal from the fruiting body to say, we were just here. Okay. And so what that means is, so we're assuming it spreads. Okay, you were just here, spread. So there's one signal. The second signal is released by the vegetative cells, which is saying, okay, we spread. Now we're moving away, directionally out. And now I have to send another chemical signal that says, oh, I'm out of food. So let me then gather together to start the forming of these fruiting bodies. And when there's high enough density of the vegetative cells. So I have to have a certain amount of vegetative cells to have the formation of the fruiting body. Okay. Next, we're going to say that the forming of the fruiting body causes nearby vegetative cells is as soon as I start to have that mounding structure, say, hey, come here. So the vegetative cells change direction to then come form and collect. And this all seems pretty responsive. And then it repeats from there. So we were thinking at minimum, there's 3 chemical responses that are necessary. And this is an open question. We don't know if that's true. Yes? We haven't done that yet. And actually, it's something that's like the very first thing I wanted to be done. But they wanted to understand, what is driving this, just the forming of the fruiting bodies itself. But yeah.
You look at the rings in the center, it looks like the math is the same. Is it a one-to-one?
No, it's not. So I guess what do you mean by one-to-one? Let me ask that first. It's not. It's smaller. It is smaller. Yeah. So because you're not producing as many spores as you did at the very beginning. Well, it's not about the spores. Your survival of your vegetative cells is not the same. So we went, instead of from this chemical response, we said, okay, let's talk about the nutrient response. And in particular, we were curious if we can capture this image from Mitch Singer's lab. And he's like, okay, you know what? This is all on his own with a bunch of other physics students. I come into the physics lab. after I was like, see if you can play with this nutrient response. And there's about 5 or 6 students trying to figure out what is the limiting behavior. Okay, I always use the word limiting behavior. I don't think this is the best mathematical notation, but this is how I think of things. What's happening at the limits? So they said, okay, well, in the first image, as we start, we know that always we will get fruiting body formation. All right? There's always a fruiting body formation at the beginning. As time goes to infinity, if the amount of nutrients go to 0, then we will have no fruiting bodies. Okay? So if the amount of nutrients is 0 and time keeps going to infinity, you're going to have no formation of fruiting bodies. If the amount of nutrients is plush, you're always going to have fruiting bodies forming. They're going to keep forming. The F represents fruiting bodies, sorry. And then finally, if we have some limited amount of food or nutrients into the system, that's what C is, representing something that we can control like a dial, then we have, again, this is forcing, right? You're forcing that periodic behavior, but this is what we're observing. So we're forcing behavior based on observation. And convincing physicists to do this is, by the way, quite hard. So they didn't quite understand, like, yeah, just choose a function that has periodicity, and we can see, is there a physics behind it, or is there some biological response reason behind it? But to start with something. That 2 to the D is actually forcing some delay of the periodicity. But he was able to do this, and this is his simulations. It's a little bit hard to see, but that green is that forming of these fruiting bodies. Yeah, so if you collect the mass of those outer rings, you'll have less than you will have at the center. And this is true biologically speaking too. So if I collect all these fruiting bodies, and this circle right here is going to be less than the center. So the nutrient one does pretty well, where we just look at nutrient dependency. All right. Okay, we have some simulations. We have something that students can play with. The light dark one is actually what I want to answer. I do want to understand, what's going on there? And that is for another student. There is a paper in the works with Mina and Mitch Singer on this. We've submitted a couple NSF grants to fund, mostly on the biology side, but they use our imagery to understand can we understand what's happening with these fruiting bodies? Under what conditions do they form? What's causing behavior? Yes. Yes. Yes, that is correct. So the simulation is the top, the real is on the bottom. Okay. So where do we go from here? Well, as I mentioned, Amina was at a Gordon conference, and one of the things that was brought up was using AI to understand this behavior, but not only to understand the behavior, but Myxobacteria actually do something interesting, is they eat other bacteria. That's their food source. And so they create secondary metabolites to eat these bacteria. And these secondary metabolites have well-known antibacterial, anti-cancer, and antifungal properties. This is pretty well established. People have been looking for these natural products for a long time. And so Nate found this really interesting paper from Wong et al. In 2023, where they were using these large language models to look for drug discovery. And so like, okay, can we do this for the species of bacteria that we're working with? And the nice part about this is we can grow this at NEIU. Okay. Our chemistry students can actually analyze the secondary metabolites that are being produced, and we can try to develop some predictive analytics of what would be if they have anti-cancer or toxicity issues. So a very good problem for an institution like NEIU. So what we did was we applied for a data supplement from the National Institute of Health, National Cancer Institute, through the Chicago Czech grant, and we got it. And one part of the grant, well, one of our aims was to do this summer research that we just did last year. By the way, NITMB is on this because at the time I was employed, and it also gave me still some time to do this work. You can see Wasal's here, and I'll talk about my students, but we have high school students that are there, grad students in chemistry, undergrads in biology, so a spectrum of STEM. So our goal, biology students grow it in the lab, chemistry students analyze it, the math, computer science, and physics students develop some machine learning model or work with existing ones to be able to come up with what the secondary metabolites are, and if we can exhibit wide range of pharmacological data. Okay, I got to sell my students because these students are all going to be graduating next year, and they're all looking for grad school stuff. So Elizabeth Jarvis from computer science, Wasal, who was doing the project during the year, and she's like, she actually is more interested in the data science side. And so I was like, okay, I got a good data science problem. And Nate, who's been a physics student, who's been working on this problem for a while. I have a little bit of time. So basically what we did is there's a database that has all this stuff in Myxobacteria. We take it down. We look for toxicity. Is it toxic, right, to humans and to other environmental factors? That's like one of the first things you do in drug discovery. And then can we take those structures and see if they have anti-cancer properties or could be successful against certain cell lines that already exist in the NIH database. The tox CSM is pre-existing, and so is Pac-Man. The students were not going to use their summer to come up with a graph neural network to do this. That is not our goal. So what we do is we look at these 5 indicators across toxicity levels. Because of time, I'm going to move relatively quickly because I do want to get it. And our goal is anything that's below 0.5. 0.5 and above is like chemotherapy toxicity. Okay. This is below 0.5. And what this is looking at is toxicity at the organic level, genomic level. And this is for every secondary metabolite that exists that we know of. And we don't know of many. But I think it's, I forget how large it is. What's the N of how many secondary metabolites that have been calculated. So not very toxic relative to like chemotherapy. Same thing here if we're looking at the stress impacts versus just basic toxicity and nuclear toxicity. These are pretty standard, but the key takeaway I want you to look at is these are not very toxic. Notice your X scales is anything below 0.5 is good. So what does that mean? Cool, secondary metabolites, if I ingest a little bit of soil out there, that bacteria is not going to kill me, okay, per se. I should be careful about that. Don't go out and eat soil if you have cancer. So the next thing is what we want to do is we want to take these structures of these secondary metabolites for the species that we're working with in the lab. And what they have is what's called a Smiley string. And what that means is you're just looking at the structure. You don't disregard the hydrogen atoms. And we compared it against 2,000 plus cancer cell lines. And we saw the following. I'm not getting too much into the weeds here. Anything below 1 means that it's effective at very minimal ingestation. So notice this frequency is like 1 on the left of -4. This is basically like just drinking a little bit of water. It's so minimal the amount that you have to ingest. Now, I'm not saying you then eat it. That's not how it works. But it's just saying the amount that you need to be exposed to, that the cancer cell needs to be exposed to, is very small. So we did this for Stigmatella rantica, and we were looking at stomach cancer line here, and we saw that we have multiple things that could fight stomach cancer. multiple for lung cancer. Again, anything below 1. And this is just one species, and there's many species of Myxobacteria. This is Myxococcus microsporus. Again, this is for large intestine. Again, anything below 1 is good. And this is for the whole digestive tract. And so what we did, I was like, okay, fine, you can give me these graphs all day, but what is actually I'm seeing? And here's some of the chemical structures. And these are the cell lines from the database from NIH. That did not come up well. And the compound names. Now, I've taken out some compound names. Ones that don't have names. There are compounds that we discovered that are not in the database. So there's no given names. Notice that the top one is stomach. At the moment, we don't have a really good way of fighting stomach cancer. And this is from Myxococcus, which are really good versus large intestine, digestive leukemia. And I said, okay, well, is there something common that you saw here? And so the students gave me these structures, and I was like, I can't read this. I literally was like, I have a little bit of chemistry background, but not enough to really understand what's going on. I mean, I was trying to look at it from a mathematics perspective, see is there some commonality of the structures? There's something that we can create. And I am going fast because I'm out of time. Something interesting happened a couple months ago. The chemistry students were actually able to detect this from some of the compounds in the lab, which is actually, we weren't expecting it to that quickly. And so what does that mean? We grew it, we were able to detect it, and we predicted it beforehand. So now, doesn't mean that I have now a cure for cancer. Obviously, that's like, doesn't happen. Okay, and that's way outside of our ability at NEIU. In terms of students, Wasal is just presented this poster last night at Abercrombs. So I'm curious how that went. Elizabeth has presented at some local conferences. And there's some even more stuff that I cannot present because biologists are very sensitive to that. So I apologize. But what have I learned? Working tangentially or working directly with biologists can be not only rewarding to myself, but also rewarding to the students. Biology, inspiring mathematics can be productive at PUIs, like quite productive. We've gotten multiple grants from NIH and NSF to do some of this work. But more importantly, for me personally, learning the language of biology is exhausting. And it could just be that in my age and my experience, but it's taken a long time for me to, and I still make mistakes, just not my language. Anyways, and there's my children. This is like my happy space is walking with my children. But any questions? And sorry for going over in time. So thanks. Yes. Yeah. So we were part of the Chicago Check, which is Cancer Health Disparities in Chicago, funded by NCI, and we had a proposal in there. So I'm using past tense, which was to actually take this and then try to then start working with mice. Because this is all something you can do in minimal risk. Let's just say that funding line. Okay, yes. So I think the next step is for Amina to be working with some colleagues at some collaborative institutions.
And you have a sense of, like, I know there's a lot of these LLM-based prediction things. Like you're not looking for a high hit rate, you're just looking for the stuff. You have a sense of, for the ones you're using, what the
It's pretty good. There's a paper that was just published a week ago that's doing something very similar, and it was really good.
60%?
I don't know what I don't have a good number. I apologize, but it's I was surprised. Um, so yeah, that's a good question. I don't know. Bill. Yes. Yeah, so that was one of my questions, right? So one of the things that happens when you're working with experimentalists is like, can you do this? Can you do? Actually, I was thinking of Alistair had his work in progress, and I asked, so he's like, well, I had all these questions. Can you do A, B, C, and D? Okay, I wasn't joking. I came in with 2 pages, like, can you try this in the lab? And one of mine was looking at that, and they haven't really taken that into account. But you saw where if they grow it on the left side, it still spreads that way. But it doesn't seem like the boundary conditions are impeding or pushing it. So that's why I have a question about that chemistry, chemical effect, because it seems like that should then affect what's happening. I don't know. Yeah.
It seems like fairly.
And people do it all the time. Yeah. Yeah.
It seems like that would be a fair question. Random research they're doing for able to sense where the nutrients are.
Yeah. I'll bring that up to Amina. That's a good question. And Mitch. No, I am. There's so many questions. Yeah. And the pattern is just fun. It's just like, yeah.
Also sort of related, not sure if you tried making multiple spots on the same regard.
So our goal was in that summer 2020 was to do that. Obviously, I could do it through these very simple AVMs, but because of that, they didn't do it. But then there's a lot of work that just came out because after that about light signal responses, and then our research kind of got deflected. So yeah, I'm curious if they've tried, because we have a lot of students that are growing stuff right now. So I will ask.
Yeah.
Yeah, like answer a lot of questions that we have too. Yeah. Yeah. Oh, evolutionary speaking, yeah, probably.
I don't know if you've seen the stuff that Alex Petrov has done at Clark University.
Yeah.
But there they see these fathom waves during the day.
Yeah. And actually, when I saw that, I was like, I thought exactly of this. Mm-hmm.
This fruiting body is somehow relates to the fact that what they're trying to feed on is also.
Oh, because yeah.
And this nice cascade of the top layer, there's photosynthesis type stuff, and then the next layer is the light on a byproduct of that. And as the light cycles move, they move up and back to respond to the light cycle.
Yeah, it probably is related, evolutionary speaking, like for them to do that, to like keep up. Okay, I give you more questions. You have now problems to play with. So thank you.